So I wanted to start by thanking everyone for joining us today. To begin, I'm going to just start with a little bit of background on Biomodels for those of you who aren't familiar with our organization. So Biomodels is a preclinical contract research organization, and we're located in Watertown, Massachusetts, which is a little bit outside of Boston. We were founded in 1997 out of the Brigham and Women's Hospital, but we're now our own independent company, and we specialize in highly translational models of human diseases and conditions for a wide variety of clients. Biomodels has therapeutic expertise within a number of disease indications, including inflammation and autoimmunity, the microbiome, oncology, and pulmonary diseases, among others. And we facilitated over 50 compounds into patients across multiple different disease indications. So just an overview of our objectives today. The overarching theme for today's presentation is to describe animal models of disease with immunological components, in which therapeutic targeting of the immune system has demonstrated efficacy both preclinically and clinically. I'm going to be talking about inflammatory bowel disease, multiple sclerosis, and rheumatoid arthritis. Andrew will be discussing allergic asthma and graft-versus-host-disease, and then Ben will be talking about immuno-oncology. So to start, I'm going to be discussing inflammatory bowel disease. Inflammatory bowel disease, or IBD, is a spectrum of chronic gastrointestinal disorders that's characterized by alternating periods of relapses and remissions. Clinically, IBD is characterized by diarrhea, abdominal pain, and bleeding, and it's most typically diagnosed by endoscopy. The etiology is generally unknown, although the environment, genetics, and the microbiome are all thought to play a role. And in terms of epidemiology, there are more than 1.5 million cases estimated in the US and over 2.5 million in Europe. These are divided equally between ulcerative colitis and Crohn's disease, with up to 70,000 new cases diagnosed annually. Onset can occur at any age, but it's most typically when young, with peak incidence in late adolescence and early adulthood. And the annual financial burden of IBD in the United States is estimated at being between $14.6 billion and $31.6 billion. Just to get a little bit more detailed in the differences between UC and Crohn's disease. UC most typically affects only the colon, and the inflammation and ulcerations are restricted to the mucosa and submucosa. Classically, Th2 cytokines like IL-4, 5, and 13 are hypothesized to predominate. In contrast, Crohn's disease can affect any part of the intestine, though it most commonly affects the terminal ileum, and is characterized by areas of inflammation that are dispersed between healthy areas. It can have extraintestinal involvement as well and can affect the esophagus and the stomach, and Th1 cytokines like IL-2, IL-12, and interferon gamma are thought to predominate. Current treatment strategies for IBD range from treating mild cases, starting with 5 ASAs, through moderate. We move into immunosuppressives like 6-MP and AZAs, tacrolimus. That moves on to biologics like anti-TNFs, and more recently the anti-alpha-4 beta-7 integrins. And then finally, this culminates in surgery for extremely severe cases. So like many disease indications, IBD is modeled using animal models. We're always seeking to have the perfect animal model, although that doesn't always truly exist. And in IBD and across disease indications, the perfect animal model will replicate the clinical condition. It will have endpoints that are translatable to both those used in clinical trials and are also easily interpretable. The underlying biology of the model will be the same in humans as it is in humans, and it will respond to similar targeted treatment paradigms. But the model also has to be cost-effective and provide actionable information. So an endpoint that Biomodels uses in its IBD models that we think makes this model extremely clinically relevant is endoscopy. It's critical that endpoints in IBD models demonstrate a drug's mucosal healing potential, and endoscopy allows us to assess mucosal disease and healing without animal sacrifice.